Okay, so good noon to all of you, and happen to be the concluding lecture as well. Okay, so I'll start where I left a day ago, and the technique part I have already explained to you. So today I'll focus on entirely the research which we have been doing for past nearly a decade. Okay. Um, and it's all about, as you can see, is hearing. Okay. So I'll tell you first how we hear sound, and then I'll tell you the questions, and then we'll see what we get as an exciting phenomena. But before I even start, let me just quickly show you what else we do in our lab. So as you might have understood now, that we mostly try to understand the mechanical phenomena in life. So in one case, we are interested to mechanically tune the tumor and then try to reverse the aggressiveness of tumors. In this case, it is a glioblastoma, GBM. It is a severe brain cancer with a survival is less than a month or something. Okay, and we are what we are trying to understand, do is first understand why or how the severity is triggered, and then if you can reverse it, so that people can still survive for more than a few years. Like you reverse it to a less aggressive tumor, which is a low growth glioma. Okay. So, in this is that, and in another case we are trying to use the force generated by biomolecules, and if you can use that and generate a similar type of motion which you get with microbes, but in laboratory system and in artificial cells. Okay. So, I always start my talk by thanking people who are all behind it. So, in this particular talk, the persons who actually are involved are BSMS students, some PhD students who have now graduated, and a postdoc who is now in UK, settled in UK, and another PhD student Nisha. Then somewhere here is Pritam and Vishav. Pritam, Vishav and Ojas you have already met, who built the magnetic tweezer, and they are the PhD students who did first part of the work. Okay. And then the financiers. So, before I even start, let me try to convince you that hearing is an interesting subject to study. Okay. Unlike any other sensors which you have used or you have learned or known, hearing is little different. Okay. It is an active sensor. By active sensor I meant is that unlike your cell phone, which has several sensors, but the sensing depends on the input signal. Okay. Your sensors in cell phones cannot amplify signal. So, sometimes I mean nowadays it is not so common. First times when the cell phone users sometimes used to climb even trees to get signal. Our ears are different. Our ears can amplify signal, and that is why you hear sound which is varying from 5 decibel to 120 decibel. It is again in a logarithmic scale, but in terms of orders there is some 7 orders of difference which we can hear, and that we could do just because our ears can amplify the signal which is coming from a mosquito buzzing a meter away where you can catch, okay, as well as the jet engine sound when it is 100 meters away. So, we can cover a huge range of input loudness, and so as the frequency 20 hertz to 20 kilohertz. Okay. Let me also just quickly tell you how ears amplify signal, but I will not go in detail, and this I usually keep because this is something which like when I was looking for a problem to work with. So, this phenomena actually motivated me to indulge myself in this problem. Is something is known as autoacoustic emission. All of your ears are emitting sound, and that is been detected when a newborn in a newborn to understand the neuronal developments. Okay, it's part of neurons. This is called autoacoustic emissions or emission, and the way it does is by dancing itself. So, what I have here, maybe if there is a sound, well, from behind you may not be able to hear the rock and roll music, but what you can see is that the one of the hair cells is actually dancing because of ion imbalance. Okay, it's not really dancing by the music, but there is an ion imbalance, and that's why they are going continuously back and forth motion, and that our ear hair cells do all the time, and they remain active even when you are sleeping. Okay, because they are always oscillating inside inner ear. So, let me take you back and explain how we hear sound once again. So, as I am talking to you, I am pushing air towards you, okay, which travels through our auditory canal, and then it hits to a membrane called tympanic membrane. This tympanic membrane is connected through three tiniest bones in our body. The names are not important, but still for the sake I will just tell the names: malleus, incus and stapes. It is thought that the big jaw which we had in our ancestors have now transformed into these tiny bones, the tiniest bones in our body, but it does a marvelous job. The stapes at the end will carry forward the vibration through this tympanic membrane to the oval window, and finally, just like hitting a drum, it will hit the oval window, okay, and hitting an oval window will create ripples in inner ear fluids. So, what you see here, the arrows, it reflects the different frequency of sound which you hear. The higher frequency sounds are sensed at the very starting of your inner ear, and the lower frequency sounds are sensed at the later stage of the inner ear. Okay. And this is the cochlea, 3.5 ton, like it goes like this way, sorry, 2.5 ton goes like this way. So, when the ripple is created in inner ear fluid, that also cause deflections on this hair cells and the hair like protrusions. Okay. Let me show you how exactly these hair cells or hairs look like. So, this is one type of hair cell, this is another type of hair cell, and these hair like protrusions are called stereo. Yes, sir.
In the previous slide, you go ahead. So, the membrane vibrates, that in turn creates.
So, it is a very so what happens is that this would so this is called tectorial membrane, and the hairs are actually touching to the tectorial membrane. So, there is a basilar membrane and the tectorial membrane. So, there is a oscillation between these two, and that causes the deflections of the hair cells on the hair cells. Oh, the three bones are just transducing the mechanical signal and finally hitting, which is something around 20 times larger in area than this. So, this is where also amplification happens, but they are taking this and finally hitting to the causing oscillations or ripples in the inner ear fluid. Okay. So, these are the hair cells, and if you look carefully, which is very difficult from behind to see, that the two adjacent hairs on the hair cells. So, this is one hair cell, okay, and there are 50 to 100 stereocilia on top, and two neighboring stereocilia are connected at their tips by a protein complex, proteinaceous complex. Okay, it is a tetramer basically, it is known as tiplings, and I am sure you can't see from behind. So, let me show it in a cartoon diagram. So, this is how it happens. Okay. As the stereocilia deflect, the tiplings would sense a tension, and in response they will pull open the ion channel, and so ions go in. This is a misrepresentation, actually is a calcium ion. The reason it is written as potassium ion, because when we made this movie, which is few years back, people knew it is a potassium, but usually the mechanosensing ion channels are not very specific. So, it is just by amount of opening, right. But later on they figured out that this is not a potassium ion channel, it is a calcium ion channel. So, it should be calcium. Okay. I forgot to change. Anyways, so now it is well understood how we hear sound. Okay. At this point, when ions go in, they change the cell polarity, they depolarize the cell, and at that point you have the afferent nerve cells which are at the bottom I had in the previous slide. Like these are the nerve cells which would take the electrical signal to brain, and brain decodes that as a sound. Okay. Nonetheless, so the proteinaceous complex is made up of two giant proteins. One is cadherin 23, which is the taller one or longer one from the top, and then protocadherin 15 from the bottom. Okay. So the question here we ask is that opening of an ion channel is just like an electric door. You press the button, it will open completely, it will wait there for some time, and again it will close. Does not matter how many people are waiting outside. Okay. So here a mosquito buzzing does that job. It will trigger the opening of ion channel. Okay. And that is how you hear the mosquito buzzing. The question now is, when I am talking to you, I am much more louder than the mosquito buzzing. What happens there? Because you see, let me take it back. So, these proteins are 2 orders of stiffer than the membrane. Okay. By now, you must have understood how what is the membrane stiffness of a cell. It is like 0.05 piconewton per nanometer, whereas these proteins would have in the order of 10 piconewton per nanometer. So, the proteins are like a very tight thread, okay, which is attached to the door, and when I am when the mosquito is buzzing, you have asked me to open to pull that thread. So, the door is open. When I am talking to you, literally I am asking an elephant to open the door. It is likely that elephant will, instead of opening, it will just remove the entire door, or it will damage the door structure, right, and that can cause cell death. These cells are non-regenerative beyond a certain age. Okay. Up to a certain age it still remains regenerative, but beyond a certain age it becomes non-regenerative. At that point, if the extra force is not dissipated, I am using the word dissipated here, which usually goes with energy, but let us say disseminated. If the extra force is not properly disseminated and the opening and closing is not controlled properly, any sound which is larger than a mosquito buzzing is potentially can damage your ears or hearing, which is not happening. So, that is the question which we ask, how these tipplings can serve as a protector, okay, simultaneously can also trigger this signal for hearing. Okay. So, we recreated the entire geometry of tipplings in vitro, and again this is not a correct representation. We also have done it on a bilayer, which is not shown here. While doing so, we also figured out the tippling structure which was reported in literature from EM based, which was long ago, was not very correct. So, as you can see here, the shorter protein, which is called protocadherin 15, if you do not remember the names, throughout I will have 2 different colors to represent 2 different proteins. This can form a cis dimer, meaning they are homodimer from the bottom also, whereas the taller one or the longer one remains independent. Okay, their hands are like this. So, initially the structure which was reported is that both are doing a handshake in this way, but we figured out that that is not the correct structure, and so we had to really go through several rounds of work to finally convince people that the original structure should be like this, the actual structure should be like this. Nonetheless, now we wanted to see how these tipplings can sense force or response to force, so that at the end there is no rupturing event happening on the membrane. Okay. Then we did the
large tippling, probably as you have mentioned several times, very hard to express in
yeah, I mean, and I never, I mean, yeah, our group never had experience in, as you was mentioning, never had an experience in throughout in my training period, PhD or postdoc, I never worked with biology or biomolecules. So, this was the first time we started working, and these proteins are too large, as you can see here. the one of them is 320 kilodalton, the other one is 120 kilodalton, and then they are forming dimers, and then you have a heterotetramer. It took 4.5 years to build this structure and immobilize them on membrane. Okay. Yeah, so those days are gone now. This part you all know by now. So, the experiment which we are doing is called Force-clamp spectroscopy. We initially start a cantilever which is far away, and one of the proteins or the dimers is attached to the cantilever, and the other dimer is attached to the cover slip. We bring the cantilever down, wait here for a certain time, so that the interactions happen, the tip link is formed. Then we pull back and hold them at a constant force, okay, to see their force responsive dissociation. Okay. And then you do this experiment several times, so that at one particular force you get a large number of lifetimes or times it takes to dissociate, and then you change the forces also. Finally, you plot their survival probability or the lifetime distribution with time and at different forces. Okay. And from there you can get the average lifetime of this complex at one particular force. Okay. Then you plot force versus lifetime. So, this is force versus lifetime, and there are, as you already know, there are 3 types of such bonds or behavior is already known in literature. One is called slip bond, which is quite intuitive. Everything which you see in real life is a slip bond. Okay. And the example I gave is just opening a pen cap from a pen. You harder you pull, faster it will dissociate, right. The counterintuitive to slip bond is a catch bond, where the bonds become stronger and stronger. Okay. So, with increasing force, pulling force, detaching force. And the third one is called ideal bond. The concept here is that the pulling dissociation direction and the pulling force are orthogonal to each other. So, therefore, the force is insensitive towards the dissociation. Okay. With this preliminary introduction, let me now show you the result. So, we did the experiment as I said. Here, unlike the cartoon schematic, which was a straight line, you will see some spikes in between. I will come back to this point later on, and you will also we will also monitor how the piezo, which is holding the cantilever, is moving in the z direction. Okay. I will come back to this point later. So, what we did was we monitored the total time it takes to unbind, and then as I showed you in the previous slide, we calculated the survival probability at different forces and then plotted the force versus lifetime. And what you, yes, right, two cadherins and 2 protocadherins. Yeah. So, only 2 outermost domains interact. I will show you that as well in later stage. And what we see is that the initially at lower force you see a slip bond, where the bond lifetime drops with increasing force. At the middle region there is a ideal type behavior. As you go very large force, you see again a slip bond, but of a different slope. Okay. So, of course, the first question we ask is it physiologically even relevant? I mean, how to understand this particular feature and relate it to physiology. So, we came up with this idea that the slip is initially the region where force is transduced to the ion channel. Okay. The ideal one is the blocker, meaning the force is now insensitive to the even bonds. Okay. So, therefore, you would expect that nothing is happening at the bottom, because even this complex is not able to sense the force. When you change from, let us say, 30 piconewton to 80 piconewton, the lifetime does not change. That means, the bonds or the interactions are not feeling that force. Okay. So, it is expected that at the bottom also the force is insensitive. So, in this region where we usually shout, I mean, this is a cartoon representation of my mom, and she is calling me in the morning, okay, which is still painful for me. Nonetheless, so in this region, when she is very angry and says, "Sunai nahi de raha hai, are you not able to hear me?" is truly you are not able to hear. Okay. and the last part where it is too loud, okay, it dissociates, so that the force is not transduced to the ion channel and should not be rupturing the system, right. So, it is kind of a seat belt, but it is a very smart seat belt in that sense. So, we believe this happens faster than it could transduce to the surface. So, otherwise I would have expected that the bottom tip link, which is now anchored to the membrane, should have ruptured the membrane or should have deformed the membrane. So, here they are getting longer time, that is what we believe.
So the changing is only on the very left part?
Yes, yes, the changing is only at this point. Yeah. So, then let us come back. Of course, the question now comes, yes, yes, it is. It retained in our.
Okay.
So, we are obtaining here calcium concentration of 50 micromolar. No, that you mean since there will be a dearth of calcium ion. So, sure, but unlikely. I mean, it is never a dearth. I mean, the 50 micromolar will never drop.
So it's always in excess in some sense.
Okay. The obvious question, how it is happening, maybe I will raise, is it fine now? Okay. Like what is the molecular reason behind it? Okay. So, first thing we studied was the force curves which we observe. Okay. Now, as I told you that I will bring it back. So, we see some spikes. What does these spikes mean? That as you apply a force, we observe a jump in the height, because this is done in a feedback loop. So, we want to maintain the constant force, okay, to the complex. And whenever there is a unfolding sort of behavior where the force is released, the piezo will move up to maintain and go back to the constant force bend, okay. And that is what you see here. So, these jumps are wherever there are spikes here, you see along with that there is a correlated jumps, okay. And when you monitor these jumps, you find one of them which is very instantaneous, okay. So, it happens, the delay in the first unfolding is very small. The moment force is applied, there is some adjustment in the protein, which is not a very large amount though. The most of the elongations happen below 10 nanometers. So, there is a short elongation which happens instantaneously the moment you apply a force to the complex, okay. That might be dissipating, but we do not know yet, okay. But we just figured out that it happens throughout the forces. There is this immediate elongation, and at higher forces you also have some higher order unfolding features at the cadherin molecules, okay. But that did not explain why the ideal type behavior anyway. So, we thought that maybe the ideal nature is coming from the complex itself. So, this was the entire structure. The complex is formed between two outermost domains of cadherin and protocadherin proteins, okay. So, we thought let us first try to figure out, like when we apply a force, how does this complex responds to this to the force, okay. So, it is a monomer interacting with just two outermost domains. Yes, sir. So, you mean this region protein can unfold also?
That can extend also by some means.
Right. So, the unfolding one was like, so I use the term as a holistically, is actually you are right. So, we do steered molecular dynamic simulations at very slow rate, and it is equivalent to force clamp. So, we do not do exactly constant pulling, but computationally also you can do a similar type of experiments. There you will see at first there will be the linkers between domains, you know, the beads. So, the structure of the protein is very interesting. You have one bead which is one domain, then there is a linker region. So, the linkers elongate. Okay. So, you can consider this as the conformational entropic structure which you are gaining at this regime, okay, at lower force and at instantaneous after the application of force instantaneously, right. Okay. So, we we do different type of simulations. The all atom simulations we use GROMACS, NAMD. That answers. Okay. Maybe I should move little fast. So, our first target was to see the feature of the binding complex, and then we thought we will also study what happens to these extra domains which are not involved in interactions. Okay. Are they contributing towards this force filtering? So, going one by one. So, we first studied the force response of the complex, same method I am not going through, and here we found that the behavior is little different. It is a slip, catch and then slip. Okay. What you observe is when you just apply the force on the complex are becoming tighter at beyond a certain force. Okay. And this region where we observe an ideal feature when you have the entire tip link. Okay. Coming to that later, now we forgot why ideal, now we were more interested to see why even catch with a single interface. Okay. And did again the simulation studies. This is easy to do all atom simulation in this point, and what we found that as you increase the force, so the simulation, as I said, it is very equivalent to the experiment. Okay. You are holding the complex at a constant force for some time, then moving to a higher force and something like that. And what you monitor along increasing force, that the binding surface area increases as you increase the force. So, this is the force here. With increasing force, you see it started from one point and now you have increased surface area, buried surface area, and also another additional peak which is coming up with increasing force. So, we wanted to see what are the amino acids responsible for for this increased binding interface. Okay. So, what you are seeing here is the amino acid interactions between two proteins and their survival. So, we do this experiment for certain time, at what fraction these interactions survive. Okay. So, there are as you can see, the purple color is with increasing force, the survival is increasing than the initial lower forces. There are several such interactions, but what was very intriguing, very interesting to us was the interaction between two amino acids. One is E78 from cadherin 23 and R113 from protocadherin, that remain intact throughout the force we apply. What does it mean? So, let us see the little more structural details of it. This is the interaction which I am talking about, which is happening almost at the center of the domains where are involved in the interactions. And what I am saying here is that no matter how strong force you apply to detach them, this bond persistence remains equal throughout. So, it is like acting like a axle. Okay. Initially the interaction was like this way, and there was an interface which was involved in the between the two proteins. As you increase the force to detach them, they are like, it is going like this way. So, this bond between E78 and R113 remains as it is. Okay. And they are working like an axle and switching from one binding surface area to another binding surface area. So, we searched in literature, are there any importance of these amino acids, and what we found that R113 is mutated to G, and that shows a phenotype which is known as Usher syndrome, which involves both hearing loss and visual impairment. Okay. So, immediately we made the mutation and studied, and we found that with the mutation the bond behavior is just slip bond. That tells us one thing, that this slip catch slip is physiologically relevant, and as you and the interactions which we found or figured out from the simulation are also helping us to understand whatever we obtain from the simulations are not artifact, makes sense. Okay. But yes, so but this catch bond we are not seeing in the tip link entire complex, we are seeing only when you have two outermost domains. So, we have reduced the tip link now, and we are only seeing the binding interface. Okay. I will come back to the entire story. So, it does not still answer why do we see ideal bond in the tip link. Okay. Then we went ahead and we increased the, so initially the interaction was just to outermost domains. Now we thought maybe the longer chain is responsible for converting the catch to an ideal type behavior, but so we made various constructs, but in every case we found that there is a slip catch slip behavior. So answer was that even if you take the single chains, it still behaves like a sleep catch sleep. The moment you bring both of them and there is certain modifications, it goes into sleep ideal sleep. So we gave up experimentally, and I went to my coffee partner Abhishek, who is an expert in simulations. Okay, so he did Langevin simulation, or this is not all atom, it is a coarse grain, and we first reproduced with like polymer beads, and we made certain connections here. We reproduced the experimental feature which we had. So, it is a sleep catch sleep. So, he modified the interactions between blue, which is cadherin 23, and protocadherin is red, is just a polymer string. Okay. And then made some interactions, so that when you apply a force to detach them, you get a sleep catch sleep behavior. You fix those parameters. Okay. Then he brought two. Okay. Now it is a pair, 2 of cadherin 23 from one end and 2 of protocadherin 15, but the protocadherin 15 is not forming any cis dimer. Okay. They are independent, and then he used the same parameter which he found here, and what he sees is that there is shift in the feature, but still it forms sleep catch sleep. And then finally he modified the protocadherin 15. Here he also connected this, which is physiologically relevant. If you remember this structure I showed you, that protocadherin 15 forms a cis dimer. So this is what it is. Okay. So he introduced few more bonds between two parallel cis protocadherin, and then he sees that the flattening of the region. How it is happening still we do not know, but at least we have theory to support that the sleep catch sleep is converted into sleep ideal sleep when you have this typical tip-link geometry even in simulation. We still do not know what changes does it do it make to convert, let me just finish this sentence, to convert this sleep catch sleep to sleep ideal sleep. And if anyone to be blamed here, then it is Shiva, because I went to him and asked him help us to understand what is the change in their mechanical features, because he can do it in very accurate measurements using his interferometer technique, but somehow it did not work yet, but I am hopeful at some point at some point we will have. Sorry, yeah, Apatin.
I think I have a reason why that bond is helping, but I will tell it after the talk.
I mean, we also have one sort of hand waving answer which says that so one is decreasing the entropy, also you are somehow having more ways to release the force from one chain to other chain. So, you are distributing the force propagation, meaning when you are pulling from this side, the force if this connection is not there, then it is continuously propagating to this point, it is also continuously propagating. So, the effective force is not released, but when you have more channels, then you are also making more orthogonal propagation site, which would effectively reduce the effective force, but entropy is also in our consideration. Nonetheless, there is no experimental validation. Yeah. Okay, so now we have everything I just spoke to you. I will move on because I am sure I am running little late. Okay. Now, in next few slides, I would like to, I would scare you a little bit, okay, by telling that every 16th person in your neighborhood are suffering from mild to acute hearing loss. Okay. If you have seen this thing, this is not our prediction, okay, WHO predicted that 900 million people would be suffering from hearing loss in 2050s. Okay. However, certain hearing loss can be addressed. Now, what causes this hearing loss? One major thing is of course noise induced hearing loss. People in Sudan a decade ago were never reported with age related hearing loss or noise induced hearing loss, because urbanization was not there. But now with increasing urbanization, there are reports from even Sudan with people suffering from age or noise induced hearing loss. This noise induced hearing loss is also common in army personals, okay. Because of the gun practice, they lose their hearing, and that is what happens. Okay. So, this is intact healthy inner ear geometry or structure, whereas this is after the noise induced hearing loss. The tip links are all broken and eventually the cells die, and as I told you, once it is triggered, there is no way to regenerate or reverse. Same thing happens with aging as well, and I am sure many of us are insensitive to high frequency sound. It goes with increasing age. Okay. I usually do that test, but today I do not have. So, what happens is that the cadherin 23, which is one of the tip link protein, is associated with age related hearing loss. It is identified as one of the loci for age related hearing loss. People did modifications in cadherin 23 and in mice, and they found that the mice undergo hearing loss with aging. Okay. So, that is how you can associate a gene to a disease. Okay. We wanted to understand what happens to this protein as we age. Fine. So, these are the now there is a problem when you try to study an age related issue, because I have a limited period to do this experiment, okay, and I cannot do one lifetime, which would be of course statistically also not quite significant, right. So, with age related hearing age related issues, you need a model which can essentially reduce this length of the measurement or the time of the measurement, still can give you a correct model. So, we looked for disease systems, and what we found that there are patients who suffer from progressive hearing loss, meaning a person who is born intact with intact hearing. So, it is not congenital hearing loss, but as the person is exposed to sound, start losing hearing at a very early age, and they start losing from let us say 8 to 12 years of age, and by 20 they are completely deaf. Okay. So, when we looked at their the modifications in their genes, which is associated to cadherin 23, we found certain mutations are connected with progressive hearing loss. Okay. So, there are several such mutations which is given here. Some cause progressive, some cause middle age progressive, some cause very aggressive progressive loss, like at 20 itself, as I said, can make a person deaf. Now, one thing to understand here is that when the person is born, the person had intact hearing. Okay. Only when the person was exposed to sound was undergoing hearing loss. Okay. Now, we are testing all these mutations, but today I will talk about a different one which was found in mice. Okay. This is not a genetic mutation, this is a created mutation in mice, and what they found that only one single point mutation in mice, which is S47-2p serine to proline, cause progressive hearing loss in mice. However, it in case of we bear S47, but then there are other variants in other species like reptiles, fish, aves, swan, where the S is replaced by V, and I mean there is no report of hearing loss with this species. Okay. And some of these turtles even live much longer than us. So, it is intuitive that the turtles will have longer hearing than us. Okay. Their hearing loss will be far delayed, not reported. Okay. It is just intuitive. So, we thought let us now use these three as our model and see how this mutant variant, which is responsible for progressive hearing loss, is having a different force sensing then the wild type variant. So, in this case S47 is the wild type for us, V47 is the wild type for the reptile and goose and all other cases. Okay. So, these are two you can say control, and this is the disease control.
So which domain of catering?
This is in EC1.
That domain also is there.
Yeah, because the number is there, na? Each domain has 100 amino acids, roughly 110 amino acids. So, you know now it is 47. So, 47th, it will be in the first domain.
So, the positions are same, but the property of the amino acid is different.
Yes,
Because serine is a polar.
yes, I agree, I agree.
Serine is a, then how that can be of similar. I mean,
So, this okay, the experiment which was done in mice, they did not control this is like random mutations, and you see the phenotype, and then there they found that this particular set of mice which has S47 to P are showing prevalent progressive hearing loss than the other mutants, right. So, this is we are taking from the disease, okay, and that is what happens. So, in this case you see the mice which are bearing S47 mutation in their tippling, there is only one mutation in cadent 23, okay, that has to be remembered. Initially they all have same hearing ability, but as they are growing in few months they become completely deaf, whereas the wild type ones become deaf at 24 months, which is typical the age of the average age of a mice. Okay. So, it is validated that the tip link, when there is a mutation, the hair cells are somewhat disturbed, and you can see the arrangement is lost at with the mutation. Okay. So, going into detail, the molecular structure wise, what does this mutation cause to the protein? You can see the structurally they all are identical. However, some of these interactions are weak when S47 is mutated to P47. So, this blue one is again shows that when you do a simulation, how long the interactions survive. Okay. So, let us say you do 100 nanosecond simulation, and then you figure out of 100 nanosecond how long these two amino acids are within a close proximity. Okay. And from there you determine the survival of these two interactions. So, you can see that in this case that is less with the survival. So, what it means that these are beta rich structures. Once the mutation is there, some of these beta strands are not having the connection, okay, or weak connection. It is not completely 0, but the connection is weaker. What can what do you expect if these beta strands are having lower or weaker connections? So, that can be again thought with a crankshaft type of motion. So, let us say if here I have the beta strands which are well connected, now you remove one of them. So, what happens here when you apply a force, this crankshaft motion which are moving at anti-correlated fashion will dissipate a small perturbation. Okay. Meaning you apply a force, you release it, because of this crankshaft connection they will come back to the original conformation. Once those are broken, then you can, there are likelihood that you will apply a small perturbation, and you would cause a permanent damage to the protein. Okay. So, we verified the same thing in computer simulations. There you can measure the anti-cross correlation among beta strands, meaning you see with the interactions how many of them are moving with a correlation and how the correlation is lost. More the dots here indicate that there is a correlation, and so as you can see in case of mutants there are certain dots missing. Okay. So,
Those motions are at really high frequency.
these are in terahertz.
And then what we are pulling is probably extremely low.
I agree with you. I agree with you. Yeah, but what the take home here is that it can deform your system.
On average it can.
Yes, it can deform. Yeah, because anyway you are perturbing at a lower speed, right, than the vibrations of this entire. Yeah, that's true. So, what we wanted to do is then we wanted to expose these 3 variants repetitively to certain exertion. Many of you or people at my age might have seen this, where I do not know why it is not playing, should. So, whole purpose is that I put a perturbation on this protein, sorry, and I see when it falls. Okay. My reference is P47. So, I would do this experiment with wild type, and the time it falls is I would put that reference from the mutant, because I know the mutant would essentially give up and would cause a progressive hearing loss. Okay. So, when the person is fallen is the mutant, otherwise the person is a wild type person. So, we did experiment, and that is why we developed the magnetic tweezer, because as I told you previously that you can exert this continuous force without rupturing the connection for a very long time, and we at first we just hold them at a constant force and try to understand the dynamics, okay, what happens to the protein. And we I will just go quickly to this part, because I have already explained to all of you. So, you can also monitor. So, what we found is that this protein of interest undergoes from the folded to unfolded state via several microstates. Okay. We wanted to determine if the one of them is the transition state or not. So, that you can do if you have these connections between different microstates, and you can see what is the final microstate from where it goes to complete unfolding, and also when it is coming back to refolding, if it is coming back through that microstate or not, but we could not find, I mean maybe we could not capture in our measurement, it was not sensitive enough to capture. Then you go more like thermodynamic type measurements. So, this is more important. What you find here is that the force range where the protein remains active, the green mark here is the force range where the protein remains active. So, what I have plotted here is the fraction of states. You have the unfolded state, folded state and intermediate states. Okay. So, what you see here, the force required to unfold the mutant is much smaller than both the wild type variant. Not only that, the width of this green color mark is also small than this and this. What this green color says, that even though the protein would go into the unfolded state, in this force regime the protein would again go to the refolded and unfolded in multiple times, meaning the protein can withstand that much of force for a longer time, for a longer force, which mutant cannot do. So, then we also, when you have this detail, you can also get the other details like cooperativity between unfolding and all. And what we found out, that V47 variant, which is again another wild type variant where which is found in turtles and other species, has maximum that shock absorbing ability, and P47, which is the mutant has the least one. We find out the kinetics, which again the same way we can find out the kinetics, we can also get the energy diagram, and what we figured out that refolding of P47 under tension is least. So, that means the protein should refold under tension and retain its configuration or conformation under a certain force, but P47, if it is unfolded once, it is very difficult for it to go back to the original state very fast. Okay. And so, once again we tested that by generating oscillatory force spectroscopy, where we are changing the force at a regular interval. Is like playing a music to the proteins, and what we figured out that, as we anticipated, that in case of S47, in majority of the times they do not fold back when you apply the next pulse. Okay. So, what it shows here is that where the folding has not happened, meaning it is going from just the entropic expansion which you are seeing here, and the percentage is much larger than the wild type variants. I think that is all. We had few more data, but I would skip, and so this is the conclusion. If you have questions,
Plus my
and the frequency sometimes or of that range, yeah, but we have scanned it across different frequency and also different force, the mean force.
At what amplitude did you get like workable data? Like what was the best frequency that you got?
No, so we collected at lower frequency because we got data in lower frequency. That's
Okay, okay.
yeah. Any no other questions? Then yeah, Peter.
Because I couldn't really, maybe it was in the dots, but I couldn't identify. But I guess, I mean, if you do these oscillatory measurements, it will be amplitude dependent, frequency dependent. And
You are very right. So I did not show where we varied the amplitude and the frequency, but we could not measure it at a very high frequency. The frequencies are all like even I mean within hertz. But we do observe. I mean, we also see damping feature, meaning so what you do is you do these measurements at a different frequency, and then you see the amplitude in the power spectrum. And if you run this for let's say an hour or so, with time, different time frame, you will see the amplitude is dropping, and the nature of the dropping is also different. Like the V40, the wild type ones actually will have over damping, whereas the mutant one is under damp. So that may be one of the reasons that it's not able to damp the input force very fast, and that's why it's actually rupturing the membrane, right? Yeah, I did not show because yeah, that's actually still not published, and this I have to share.
The same construct I think we have, the mutation that you have sent us.
Yeah,
We are
so
measuring stiffness directly of the mutant versus the wild type.
right. So what the constructs now we have, that in one case you can have the entire like cis dimer and then cadmium 23, and forms the original tip link. But then we made a mutant so that we can break the cis interactions. So still dimer, but the connection is now lost. And what is the difference in their force response? That's lying in his refrigerator.
We have that. So the S47.
Yeah, even all these three also you have. Yeah,
Yeah.
yeah, Sanket. You see, they may be lying at this way. But if you see this way, the force is also, the tension is also feel like this way. So eventually they are linear, right? Force is also, tension is also between these two, na. So always at one end and one end, which is linear, right? So either way you do experiment this way or this way, both will give you same result, है ना? The force estimated should be correct. After that, I mean, it's collinear. So the force estimation says that they withstand 10 piconewton to roughly 100 piconewtons. But they are of different heights, na. So this bigger one will move like this way, the shorter one will move like this way. So always the force will be along this axis, na. So tension is along the fiber, and you are yeah, I mean, however you see this way or this way, it's same.
Time for some concluding remarks. Some of you will say a few things. I think let me first invite Peter to say a few things about this whole course. And
yeah, first of all, thank you very much, Shiva, for setting this very nice course up, and also for inviting me. And I have to say I enjoyed it really a lot, and I also wanted to thank you for these long attention times. Yeah. And I was really impressed by the questions that you also raised during the talk and after the talk. So you were very active, and that was really very good. So I enjoyed it a lot, and I want to thank you also for your contributions during this week. Thank you.
then
I have nothing new to say. Yeah,
it's customary to say a few concluding remarks. So,
this is a very odd situation actually. Yeah, I mean, I hope you all enjoyed, and as Peter said, we from our side we also enjoyed a lot, like interacting with you in person and also in the tutorial and the teaching session. Yeah, thank you.
yeah, and
Yeah. I mean, I had a few slides, and but if you, I also wanted to say, whenever you can very easily drop me an email if you have whatever questions you, any kind, welcome to send me an email, and if you prefer a little bit of a cooler temperature than what you have here, then I recommend you to come to the Linz Winter Workshop and Linz Winter School in January.
Thanks, Peter. So I guess the aim of this Gyan course was to introduce you to AFM, but also we got introduced to magnetic tweezers. There's another sister technique called optical tweezers, and I recommend you go through all these techniques and do the comparative study on your own. Now, if any one of you seek advice on AFM data interpretation, some technical help, whatever, I am there, Sabisach is there. Drop us a line and we'll be very happy to assist you. Through discussion sessions, we have seen kind of problems that you have in mind, and they're all exciting and fantastic ideas that you have in your head. If you need any help, getting results or getting interpretation or whatever, please do not hesitate to drop us a line, talk to your advisors, and also we are free to do Zoom chat or whatever is appropriate. Okay, so now any one of you want to say something, quickly? You were asking a lot of questions. You have to say something.
So I enjoyed a lot this course, and I for my colloquium also I have read this book, Single Molecule Biophysics, and I didn't know that this was written by Peter himself. So personally knowing him and get to know about his work and also about you all. So this I have enjoyed a lot, and yeah, I've asked questions because all those sections have intrigued me a lot. So thank you a lot. Thank you.
Anyone wants to say something more? D- Lunch is still about few minutes away, so don't worry.
So you are not missing anything.
Saket, you want to say something?
Yeah, it was fantastic course, I guess. I also, as he said, I had read Peter's paper during my molecular sensing course that I taken in my institute, and I reviewed it and wrote the term paper. That time I didn't understand many of the things, but attending this thing, I got a really very broad and deep perspective of single molecule force spectroscopy and where it can get applied, And other than that, the instructors were fantastic. They gave lot of time, lot of ideas which we could discuss with them and get their inputs and feedback on it. And yes, I think it's a great platform that has come up to bring bio-SPM to a broader community. So it was very nice. Thank you so much to all.
So there to add, even I told the same thing to Peter, that technically you are my teacher as well, because I learned doing AFM and doing force spectroscopy measurements by reading your papers.
That's great.
I learned very late, but yeah, his papers like phenomenal.
Yes. Okay. Some of the girls will have to say something beside you. Okay. Good.
The course was really nice, sir. Yes, yes. Okay, so the course was really nice. We got to know a lot of things. Like every doubts I had in my mind, this was my first time like knowing about AFM. So now I can say after 5 days that I know AFM pretty much. So I can do my sample analysis by myself. I may try that, and if not, then I may like contact you anytime. So we have developed a community that is there, and there are no such courses that actually talks about research. So it is very nice to talk about research here, because in general in educational courses we only just get to know the techniques or the basis of it, but how to actually implement it, it is a really nice initiative. Thank you.
Just to keep the balance, one of you.
Okay.
You can come.
Yeah.
It's okay.
Um, so I come from a very developmental biology background, and I've done a couple of AFM experiments with the help of the people in the chemistry department. But actually understanding what goes behind your experiment and how it's analyzed, I think that's something that this course taught me. And I'm very grateful to all the lecturers and the instructors who took out the time, the energy, and the patience to answer all our questions. And I'm sure this is going to help me in my PhD project and future. So thank you so much.
Okay. Good. You want to say something? Come in.
Everyone has said everything. I don't have to say anything. Just last one word that I am very much thankful to all the volunteers over here, cameramen, and all the non-teaching staffs also, so that they have made this possible in a very quite polite manner, and that to be very fresh manner. It is not like that ki everywhere we can see a lot of dust or there when we go to some other conferences. So they have done a very good job. Thank you so much.
Okay, so I think let's give a big round of applause to all our team of volunteers, which are PhD students from my lab. They really have taken time and effort to manage this smoothly. Many of the things for instance, certificates I had never had a look before, and they have come out so well. When he showed it to me, it is amazing the kind of efforts my research students have put in. So